we've looked at how the various ways we can work on the DNA. You learn about cutting and joining of DNA. You learn about inserting the DNA into whole cell and learn about selecting cells that have been transformed or transgenic or GMO. You learn about using gene probe to fish for the gene that you want, and hence there's application in microarray to look for the genes present in organisms and also to look for genes expressed in organisms and the degree of expression. So all these are the techniques that have been developed, and therefore we started off by looking at application of this technique in medicine by looking at previously the advantage of using recombinant proteins to treat diseases, right? So for today, we're going to look at another technique used, basically is actually DNA probes that are being used for genetic screening. So the probe will be used to screen for, example, breast cancer. So screen for probe, DNA probe is used to screen for complementary DNA, right? So it's used to screen for alleles that code for, which can result in breast cancer and Huntington's disease and cystic fibrosis. So we can use the probe that can detect the mutant allele that result in these diseases to find out whether the chances of the person getting this disease before the onset of the disease, so they can do something before that. So prevention is better than cure, or preparation is better than suddenly strike with that disease. So genetic screening is the analysis of an individual like us, or the fetus, or the embryo's DNA, to check for the presence of a particular allele that result in genetic disease. So therefore, to check for the presence of allele, we have to use gene probe, right? Because you got all the DNA found in the nucleus of the cell. So these are all the DNA or the chromosomes. So among the chromosomes, we want to know whether there's presence of an allele, for example, that code for hemophilia in this person. So we have to use a gene probe that's able to bind to the allele that causes hemophilia, for example, bind to this one. So you bind to this one, it means this person's DNA got an allele for hemophilia. So at least there's one allele for hemophilia. So that's what we want to do. So these are the 3 examples stated in the syllabus that you need to learn. The genetic disease that we can screen, which are breast cancer, Huntington's disease, and cystic fibrosis. So we know that these diseases arise because of mutation of the normal gene. So when the normal gene is mutated, the base sequence is slightly different already. So it results in a different allele. Means alternative form of a gene, right? Once it mutated, it's no longer the same base sequence, it's alternative form of gene, so it's a mutant allele. So the normal gene for breast cancer is BRCA1, BRCA2. So BRCA stands for breast cancer. In actual fact, this gene is a tumor suppressor gene. It's supposed to prevent tumor from forming one. Okay, once there's mutation to this gene, therefore it cannot code for a functional enzyme which can correct the mistake and therefore repair the DNA, then it will result in breast cancer. Because cannot stop tumor from growing, because cannot repair DNA. So cannot stop the cell from growing. Then for Huntington's disease, the normal gene is HTT gene that code for a protein called Huntington. So you have learned that when this gene is mutated, where when the repeated sequences becomes longer, it will result in a product. Huntington is slightly different from the normal one. So this results in Huntington's disease. This one you've learned already, correct? Okay, you've learned already. So cystic fibrosis, normal gene is CFTR gene. This gene code for carrier protein in the mucus secreting cell. You're not writing notes, So that protein, the carrier protein, is called cystic fibrosis transmembrane regulator. That means it's located in the membrane, across the membrane. It's supposed to transport substances across the membrane. So what is cystic fibrosis? It's a kind of disease whereby the mucus of the person becomes very thick. So when it becomes very thick, it sticks to the airways and it's very difficult to cough out. So this person got problem to breathe because the airways are always being clogged by the mucus, which is so thick that you know, sometimes when the mucus is very thick, you cough also cannot come out, right? Because it's very sticky. So they have this problem, so they cannot get enough oxygen to their body, through their lungs to the body. So they're always breathless and always very weak because all the cells in the body cannot get enough oxygen. And this person may die if they cannot get enough oxygen. Okay, so genetic screening. So there are 3 targeted individuals that we can do genetic screening. First one is carrier screening. Second one is prenatal screening. Third one is pre-implantation genetic diagnosis. First one is carrier screening. Means for normal people like us, we want to screen whether we are carrier of a certain disease. Okay, we are carrier of the allele of certain disease. Prenatal screening means pre means before, natal means birth. Before birth, you screen the fetus, you screen the embryo, which is growing inside the uterus of the mother. C, pre-implantation genetic disorder is before implanting the IVF embryo. Okay, that means embryo that's been produced by fusing the sperm from the father and the egg from the mother outside their body in the lab. So embryos are formed. So we want to do a genetic screen on the embryo to see whether there's any genetic defect or not, so that we can choose an embryo that is normal to be introduced into the mother. So this is the advantage of doing IVF. You can choose. Okay. That means, can you choose the gender as well? If you do IVF, can you choose the gender? Can, right? Is it legal to choose the gender? Yes or no? You can choose your first let's say your first child is a son, right? Then the second child you do IVF, you can choose a daughter, right? Can. Theoretically can. Practically, whether it's allowed, I'm not sure. Okay, I don't know. So maybe different countries is different, huh? Okay. So the next thing is, so once you've done the genetic screening and we detect that this person contains this mutant allele, which can result in the genetic disease, what's next? Next is you need to know the patterns of inheritance of this genetic defect. That means whether this allele that caused the disease is a dominant allele or recessive allele. Say for example, let's see here. Now, Huntington's disease coded by dominant allele or recessive allele. Hello? Huntington disease dominant allele or recessive allele? Who said dominant? Who said recessive? Hello, it's dominant. Huh? This better go and revise, okay? Huntington disease is coded by dominant allele. The only thing is that the onset of disease is later, but doesn't mean that it's not there. So let's say, for example, we are checking for screening for Huntington disease, and we discover that this person has got heterozygous. So when this person is heterozygous, because this Huntington's allele, right, is a dominant, will this person got Huntington or not? Huntington's disease? Will, isn't it? Because dominant, right? But if you're talking about hemophilia, hemophilia is coded by dominant or recessive allele. Recessive, that means to say that this person has got homozygous recessive, right? Like this one here. This person must be homozygous recessive, okay, to actually show the disease. But Huntington's disease, any one of this genotype will give the disease already. So after you've discovered, then you ask yourself, so this genetic disease, is it coded by dominant allele or recessive allele? And then if you screen this person, is this person homozygous or heterozygous for that allele? Then you'll know that whether this person, what is the risk of this person developing the disease. And then you also need to know whether it's sex linked or not, because sex linked also affect how this allele is inherited to the next generation. and whether it will be shown in the phenotype in the next generation. Correct or not? Let's say, for example, for the son, right? Among these two sons here, right? Only this one will get hemophilia, whereas the one on the left side will not get hemophilia. So you also must consider whether it's sex linked or not sex linked. So when you want to know the information, okay? So we're going to go through the screening for all these three groups of people. The first one is carrier screening. Carrier screening can be the first one is we screen all individuals in the family when we suddenly discover that someone in the family developed, for example, breast cancer or developed Huntington's disease. So when someone develops this disease, we want to screen everybody to find out whether you are the carrier. Because, for example, Huntington's disease, if you're a carrier and it's a late onset, you may not know whether you have the allele, right? And then somebody in the family got, so it runs in the family, so you want to do the test to find out whether you have the allele. So why do you want to know whether you have the allele, even though you're normal now? The disease may develop in this person, isn't it? You're probably 40 over years old, 50 over years old. So then you can look at your life differently, you live differently. Correct? Okay. So that's why you want to do that. Okay, that's part one. Part two, screen potential parents. So potential parents, when couple, they want to get married and they want to have their own family and children, then they know that their genetic components will be passed on to the children. So they want to know whether they're carrier because both of them look normal. But when they marry and have children, their genetics will be passed on to the offspring. They want to know whether they have the defective allele, and when it's passed on to the offspring, what is the chances or risk of the offspring getting that disease. Okay, you want to know. Then after that, you can make decision, isn't it? Informed choices. So let's do a case study, because this chapter is about benefit, advantage of genetic screening. It's focusing on advantage. So we want to know the advantage. After I know what, so what? Okay, after I do the screening, so what? So first case study is about Huntington's disease. Huntington's disease is coded by dominant allele, capital H. Okay, so the question is, what will you do if you discover that you are a carrier? Someone in your family at the age of, let's say, 48, developed the symptoms for Huntington's disease. And the doctor diagnosed that, yes, this person actually got Huntington's disease. Then after that, the whole family go for screening. And you discover that you're a carrier. So once you discover your carrier, what can you do differently? What will you do differently? Megan? Don't care. Just live until you get Huntington's disease. You? Same. Don't care. Just carry on. Then after that, when you got Huntington's disease, you cannot think properly. Then after that, how are you going to decide? My house, this house give to who? This money give to who? This car give to who? Or everything give to government? Decide first. You must write your will first, ma. After you wrong already, you cannot write your will already, how? Correct or not? Then are you going to search for places that will take care of this kind of people so that at least you look for it and you plan your financial planning so that your money is allocated for that purpose once you become wrong already? Can or not? If not, by the time you get wrong, you got no money, then everybody come and take away your property lah, your money lah, then after that, you left you at the roadside. Correct or not? So you can make decision once you know this. For example, it gives you early diagnosis. You know that you have that allele, so you've got very high risk of developing that disease. So early diagnosis allow you early prepare of oneself before the onset of the disease. For example, what do you mean by prepare yourself? You write your will, you write your will when the brain is still working. Then the decision is you can decide who to give what to who. Correct? Okay? Financial planning to support oneself before onset of disease. So you plan your money rather than every time, never mind, life is so short, whatever money I earn, I just use it. I got friends like that one. When they're young, they can wait until they old and see. When they don't have much strength to work and get money, and then all money all used up already, that's it. Okay. C, research on NGO. So you can start to research on the non-government organization, groups, facilities that can support people with Huntington's disease. So that when the symptoms develop, you know where you can look for support. So these are the things you can do, isn't it? Okay, so these are the benefits. Now, case study number 2. Let's say, for example, next time, who wants to get married next time and have your own children? None of you are.
Just 19 years old, I think so far.
Yeah, it's not in your field of thought yet, right? Okay, so maybe some of you will, some of you won't. Okay, so what will you do? Let's say this couple wants to get married and have children. Then they went for the genetic screening and they discover that both of them are carrier for hemophilia, say for example. Then after that, the genetic counselor, once you do genetic screening, the genetic counselor will help you to read the results and make decision. So say for example, you got the result already, you see among your children, this is not hemophilia, this is something else. Say for example, this is another disease which is recessive, like for example, cystic fibrosis. Let's say cystic fibrosis. So you look among your children here. What is the chances of your child having cystic fibrosis? What is the risk? How many percent? Based on the diagram, how many percent? 25, isn't it? That means every birth that you give rise to, then there's a 25% chance that the baby will have cystic fibrosis, you know. So because of that, the question is, what will you do as a couple after you get this information? I give you the choice. Never mind. Follow the natural course of pregnancy. Will you choose this? Or, I will choose IVF and do embryo biopsy. I choose embryo that is normal. So I choose the normal one, this normal one. Can or not? Or I adopt a child. So which one will you choose? How many of you will choose A? Hello? How many of you will choose A? Please raise up your hand. No A. B? Two of you. Very good. Three. Okay, good. B. Okay, then the rest of you C. Yeah. The rest of you?
Just option B. You got option B.
Option D was abstinence. Abstinence means don't have sex. Then you got 0 chance of getting hemophilia child. Which one you choose? A, B, C, D. Either A, B, or C, or D. Oh, don't get married, that's it. Or get married, don't have children, that's it. Hey, very difficult to know, because the moment you get married, right, at that time when you get married, you say, I don't have children. But after 5 years down the road, you see, who are Andrea's got 2 kids already and then who else got 3 kids already and generally got 1 son already then suddenly like you totally that I also want to have, I don't want to have, like that it's impermanent it's nothing. Your decision now is not permanent I tell you things will change with time.
What do you feel?
What do I do
What do you feel?
How do I feel?
Yeah.
Feel what?
If you don't have children.
I feel very free. That's why I say there's no right and wrong. It's how you want to live your life. There's no right and wrong. There are people who enjoy taking care of children, even though it's like putting a lot of time and effort. They enjoy the process of bringing up the child, and they feel very fulfilled. Some people, they feel like hell, when they take care of children. All my money go there, after that, kick some, kick C more. Okay? So very different. I cannot say right or wrong. It's how you handle the things only, how you look at it. Correct? For me, I'm very happy. That's it. Okay. Freedom. Okay, next. Right, case study number 3. Let's say, for example, someone in the family, like auntie in the family got breast cancer, and then the grandmother also got breast cancer one, Great grandmother also got breast cancer, So it's like, oh no, it runs in the family, okay? So therefore, you go and do the breast cancer screening to look for the mutant allele for this cancer gene BRCA1, BRCA2. After screening, ayo, present or how? I forgot. Got the allele. So how? Mutant allele, no? Okay, tell me. This for the female here, okay? For male, if your wife got, then you tell me what you do, or your girlfriend got, okay? What will you do now if you got that information or you're present? No? How? Use a knife and cut away your breast. See whether you want to cut. You cut your finger also already. How to cut? Okay. Do you want to adopt a healthy lifestyle which will reduce the risk of getting cancer? Right? You may have the allele, but it doesn't mean that it can be activated because it depends on a lot of other factors. Whether your cell is healthy, right? Take the food that is more natural. They don't have a lot of chemical additives that is carcinogenic. Right? Take the drinks that are healthier, not those with a lot of chemicals, coloring, and sugar. They say sugar is one of the cause of cancer. Why? Cancer cells grow very fast. What do they need? Sugar. Okay? Need lots of rest so that your cell can have time to repair itself, right? Especially DNA, mistake, right? Error in DNA. When you sleep at night is when our body repairs itself. Do you allow your body to have enough rest or not? Stress also, handling of stress. There are research that shows that people who actually have got cancer, right? Majority of them, right? They have gone through a period in their life where they have a lot of stress, emotional stress, and after that, cancer starts to grow. So you can actually relate because when the person is stressed, the body's condition is very acidic, very tense, not enough oxygen. And then that kind of condition actually allows cancer cells to thrive very well. So you can see that a lot of factors that can lead to cancer, not only because of the allele. You have the allele doesn't mean you will have breast cancer. So do you want to choose a healthy lifestyle or option of breast removal now? Faster, make appointment, doctor, remove it. No breast cancer. Full stop. So this is called elective. That means you choose mastectomy means remove the breast before cancer grows. So you choose A or B or C. Don't care. When the time comes, you will scold yourself. You will go and look back and scold your younger self. Why you didn't take care of me? Now I have to suffer because of you. Because why not? No wisdom. Follow the mind you follow the mind as not wise. The outcome will be disastrous. Understand? You want to follow a wise man that show you the direction, or you follow what crazy man? Not crazy, I say that unwise Dumb, not dumb dumb plus unwise They give you idea that is not beneficial for you you follow that idea and see then you see. Okay, right. So sometimes you have to be very firm when you face those kind of idea. You got to be very firm when you got this kind of idea that tell you that never mind it's okay life is cushion enjoy the life Remember I tell you all before, life is not about, I think I didn't tell you, I tell the other class. Life is not about doing what you like and not doing what you don't like. Hello? You're suddenly shocked already, Life is not about doing what you like and doing what you don't like. Because doing what you like, you think what you do is beneficial for you or not, or disastrous, you better think twice. So that phrase is not correct. That phrase is leading the whole society to the wrong direction. You need to check, when I do this, is it beneficial for me, suitable for me or not? Then you are putting, when you let out the animals, they run very well everywhere, okay, and they hit everything everywhere. But when you put what do you call it, the fence around it, this is where you can roam around. This is a safe place. That is not. Don't go out there. That you actually protect yourself from unnecessary problem, because you choose a decision that's not suitable for you, and the outcome is actually not so good. Like for example, study already you choose to study or not to study, you choose to do or not to do, then the outcome you know already. Then why you know, then you still choose that? Because you follow the mind that says never mind lah. I'm still young. It's honeymoon year, college, chill, you know. And then after that, yeah, YouTube is nicer lah. Why you do this thing that crack your head ah? You know. So you follow this master and pray to this master. You make this master very fat, and this master what now will govern your life already. Because why you invite him to come and sit in your house and then make coffee for this person, make tea for this person, bring all the what, bring all the cakes and then they become very fat there then they govern your life rather than you master your life. You see or not? Sometimes you don't see this one or no. Okay. Right, next. This is about genetic screening, right? And psychology. Okay, next. Now, this is just like talking about screening individual, right? Whether they're carrier in a family or a couple that want to start a family. Now, what about, they already started. They didn't know they can do prenatal screening. They didn't know they can do a carrier screening. So after pregnancy, then only they want to screen whether fetus is normal or not. So prenatal screening, there are actually 3 ways. Okay, 3 ways. First way is chorionic villus sampling. So this embryo stage. Second way is amniocentesis. When the fetus is very big already, we don't want to touch the fetus. We only take sample from the amniotic fluid. Third way is the best way, non-invasive screening. This one will not have risk of miscarriage. Number 1 and number 2 requires needle to be put into the uterus, and therefore there's a chance of injuring the fetus or embryo, and there's a chance of miscarriage. Third one, nothing inserted into uterus during pregnancy, so non-invasive. So first one is called chorionic villus sampling. Now, chorion is the outer layer of the embryo at the early stage of embryo. Now, you know that first, after fertilization, a zygote is formed. So this is zygote. Then after that, the zygote will divide by repeated mitotic division. So you will form embryo, right? The embryo, the size of the embryo is still similar to the size of the zygote because the cell only divide but did not grow in size. So you got the same structure size, but then you got a lot of cells inside it because of repeated mitotic division. Okay? So we call this morula, which is a ball of cell, right? Morula. And this is actually embryo. After zygote, all the structures here are embryo until you can see the limbs are the shape of the organism is called fetus. Before fetus, they are all embryo. Before fetus, they are all embryo. Other than zygote is zygote. Okay? So morula, all the cells here are not differentiated yet. So you can pluck any of the cells out from here and do a test. It's perfectly okay. Because they're not differentiated, it will not affect the growth of the remainder cells. After that, it will differentiate, okay, into this structure already. Okay, so for one of the stages of embryo growth, right? Like this one here, after they differentiate, the outer layer, this outer layer of cell, this outer layer of cell is called the chorion. Chorion is the outer layer of this stage of embryo. So because it's early stage, it's still embryo. So if the doctor want to take the cell out from this, the cell out, they will have to take the cell from the chorion. Okay, so this chorion is the outer layer, which will not form the fetus. So therefore, you see, there's the ultrasound scan that scans the uterus, and then there will be a catheter that is inserted from the vagina through the cervix into the uterus, and then try to take a small sample of cell out from the chorion layer. This chorion eventually will grow to become placenta. Okay, so that's why they said early placental tissue, which is actually the chorion. Okay. So this is how to take a sample, and this is done at the 10 to 12 weeks of pregnancy, which is about 2.5 months to 3 months pregnancy. This procedure is being done for genetic screening. But if it's later stage of pregnancy, like 13 weeks to 18 weeks of pregnancy, then the fetus will be formed already. So therefore, the doctor will do an amniocentesis means you try to withdraw amniotic fluid, okay, out from here, and then test for the DNA, test for the allele, the mutant allele. The reason is because when the fetus is growing inside the amnion, right? You know our skin will shed all the time, right? So the fetal skin will also shed. And then do you know one thing is that the fetus will urinate inside the amniotic sac. So the urine of the fetus will go into the amniotic fluid, and when it urinates, the cells on the wall of the urethra, where the urine come out from, it will also shed. So all these cells contain the DNA of the fetus, right? So we just withdraw the amniotic fluid, which contain the cell of the fetus, and then do the genetic screening. Okay. So this amniocentesis. Now nowadays they got a better method, which is a non-invasive prenatal screening. Non-invasive prenatal screening is where they will take the blood from the pregnant woman and then test for the presence of the allele in this fetus. The reason is because, you know, as the fetus grow for 9 months, right? Some of the cells may die off as it grows. And then you see here in the blood sample, you've got mother's cell, so maternal cell, and you've got the fetal cell in purple color. And then some of the cell already die off, but the DNA, the proteins will be spread all over the blood. So it'll be inside the blood. So therefore, they can take a sample from the mother, which may contain the DNA fragments of the fetus. And then they can test for the presence of the mutant allele. So this is the latest method, okay, non-invasive prenatal screening. So it's a safer method. DNA fragments from the placenta cells that have the same genotype as the fetus find their way into maternal circulation. So do you know just now I said already placenta grow from the embryo one? So the cell in the placenta is similar to the cell in the fetus, isn't it? Similar, okay? They come from the same source. They come from zygote, right? So they're genetically identical. So they just take those cells from the placenta that have gone into they don't take the cells, sorry, they take the DNA, DNA from the cells from the placenta that have gone into the maternal's blood. So fetal DNA fragments are isolated from maternal blood sample and tested for. So what do we test when you do all the prenatal screening? 2 things we test: chromosomal mutation, okay, and specific gene mutation. Only 2 things. Chromosomal mutation means we want to know whether the number of chromosomes is correct, 46 or suddenly 47. Trisomy 21, so it's become 47, right? Or some of the chromosomes become longer or shorter. Because when the chromosomes become longer, you got additional genes, right? Become shorter, you got less genes. The genes supposed to have, you don't have. Then there'll be abnormality in the fetus, for example. Gene for eye color don't have. Eye become what color? No color. Okay. Specific gene mutation means you want to look for cystic fibrosis, Huntington disease, all these things. So you have to use the probe for that mutant allele to see whether this fetus got this genetic disease or not. So this is a case study. So you need to make decision. If the fetus, let's say you are the parents and the mother go for prenatal screening and they discover that the fetus has cystic fibrosis, what will you do as the parents? So cystic fibrosis is it gives a symptom whereby the mucus is very thick, it clogs the airway. So anybody with this cystic fibrosis have difficulty breathing. I give you the full story. They have difficulty breathing, so the doctor have to give them enzyme to break the mucus molecules into small chains so that it becomes more soluble and the mucus becomes less thick, so it can easily be coughed out. So the baby, once it's born, right, you have to quickly give them this enzyme. And also because the mucus is inside, it's always trapping all the bacteria, fungus, viruses from the air, right? Once go in, stick to the wall, right? So stick to the mucus, isn't it? Then it can start to move around and infect those cells of your airways, and also the lungs. So you have to let the baby breathe in antibiotics, because we want antibiotics to go to the lung. So to breathe in. Inhale in the antibiotics, inhale in the enzymes. right? And also there's another therapy, is a physiotherapy. So the physiotherapist will rub the back of the patient. I don't know how they do it, so that it can loosen the mucus so it can easily come out. So these are all the treatments that's required for this person for the rest of their life. Okay, so imagine your baby will have to go through this one for the rest of the life, which needs money to buy the enzyme, right? Money to buy the antibodies, money to pay the doctor, right? Money for physiotherapists, all these are money, dollar sign, money. Okay. Then mental stress, when you see other people's children, wow, so active, go for swimming competition, don't know what thing your child cannot walk. How? So I mentioned this one. What will you do? So you need to source for early treatment, so that if you do not know these things happen, the fetus may die, right? Let's say you do not know this fetus has got this illness. Once born, it may die. Right? You got difficulty breathing, you still don't know what happened. After that, late treatment and late diagnosis, then that's it. So once you know, you can prepare already. Once it's born, you put them under all this treatment. Okay, early diagnosis, early treatment. And then prepare for long-term whole life treatment costs. So you need to get ready the money for the whole life treatment. Okay, the cost, where the money come from, I don't know. You have to think about it. Okay. Source for financial aid. So if let's say you really cannot afford then is there any financial aid from the government? Do they allocate any special fund or not for special children? I think for like Down syndrome or this, government actually provides special fund. Okay, so government provide financial aid or not? NGO, donor, got or not? Okay. And also you need to source for mental support and emotional support from group of parents group of parents with cystic fibrosis children. So they come and support one another, how they face the challenge, how they overcome it. Okay, so you got this group. So you have to start to look for this kind of support, financial, emotional, mental. Okay. So that group of people that will get this genetic screening is actually the embryo for IVF. Okay. So you see, this is actually a ball of cells. Can you see that this is actually embryo in a ball of cell stage? It's called the morula, 8 cell stage. 1 become 2, 2 become 4, 4 become 8, right? 8 cell stage. So at 8 cell stage, all the cells are not differentiated. At that time, you can actually withdraw one cell out from the morula, which is the embryo, and then do the genetic test on it. So this is called embryo biopsy. Okay, embryo biopsy means taking a cell out to test for this DNA. So if you discover that this embryo has got the defective allele, we will destroy it. We will not introduce it to the mother. So this is called pre-implantation. Genetic diagnosis. Okay. So this topic is basically learning the advantage of doing genetic screening. Whatever that's listed here, I already go through with you just now. So I do not see there's a need to go through this again. You can read it on your own. Once you understood it, you can just read it on your own. It gives you early diagnosis. It allows you to prepare for early treatment. And for couples, you can decide whether to have your own children or to do IVF and embryo biopsy, or to adopt a child. And for late onset, for example, your carrier late onset for Huntington's disease, you can make early preparation, like write the will, source for support center, that when the Huntington's develop, you got a center to support you, and then plan your financial so that you have enough money to spend when you can no longer work. Okay, so all these things are advantage of doing genetic screening. So these are all things that I've mentioned just now already. So you can read on your own. All this we covered just now already. Okay. One thing is this one is interesting. Can reduce economic cost to parents and country to keep a child who is suffering. Just now, did I say abortion or not? I didn't say abortion, right? Let's say, for example, you're pregnant for 3 months already. Suddenly you realize that the fetus got Down syndrome. Right? Because just now got do prenatal screening, right? I didn't give you case study. Prenatal screening study, got Down syndrome, 3 months down the road. So you as parents, the father and the mother, hello father. Okay, so the fetus is Down syndrome. So how? What's the decision? Continue until full. Follow natural path. Or you decide abortion. This is called therapeutic abortion. Therapeutic means something wrong already, so that's why you want to treat it, so you abort it. So it's called therapeutic abortion. Or prepare to have it and then get a lot of information about the support that's needed, the financial support that is available for you, and be emotionally prepared. So which choice will you make? First, second, third. Who choose the first choice? Continue to have it. Who will choose therapeutic abortion? Who will choose continue to have it, and then after that, we'll look for sources to support you? Wait, those of you didn't raise your hand. Difficult decision, right? Now see, if putting you into that space and it's difficult to make decision, that means, should we go and judge people? No, we cannot judge people, and we cannot say it's right and wrong one, It's like whether you're mentally prepared, you're emotionally prepared, and you're financially prepared to support this child, Sometimes a lot of money is involved, right? Are you financially prepared or not? If you're not, why you want to have it? Just because people say that, this is called killing. You kill mosquito or not?
Sure, very different story.
You kill cockroach or not? That's killing also, no? Are there? So when it comes to this kind of decision, right, you cannot say this is right and this is wrong already, because we do not know the background of this person, the background of the family. Are the family members supportive or not? Are the parents in good terms with each other or not? Do they have enough money or not? Do they have enough support? All these things we have to think of. Are there any available treatment in the country or not? Are the medicine expensive? Are the treatment expensive? All these things must take into consideration before you make decision. So there's no such thing as, you're very cruel, you do abortion, No such thing one, because you're not in the person's shoes to really understand all the conditions surround this person that makes this person to make this decision. You don't know one. So we cannot judge people. Really cannot judge people. Okay? So when they make their choice, that's the best choice for them, whatever condition they have, which we will not know, right? I will not know what's the condition behind you. I'll just see you here, right? Right? So I don't know. I cannot judge. Okay? So I see today's this topic is very good for you to learn a lot of things. So let's recap. You have learned the definition of genetic screening, where you want to use especially make gene probe to detect mutant allele that give rise to genetic disease. Okay, and then 3 targeted groups for genetic screening. Number 1, the carrier, right? Either in the family someone got the disease, so you have to check whether you're carrier, or couple planning their family, you have to check whether you're carrier. So what is the chances of your children having it, and then you can decide. Okay, this is called family planning. Then number 2 is prenatal screening of the fetus and embryo, and number 3 is pre-implantation genetic diagnosis. Okay, 3 targeted groups. And methods of genetic screening I've shown you for the prenatal screening. Okay, you've got what? You have got amniocentesis, you have got the chorionic villus sampling, and you've got non-invasive genetic screening. 3 ways. Then examples of genetic disease and mutant alleles to be screened. There are 3 diseases that we mentioned, right? Breast cancer, BRCA1, BRCA2 gene that are mutated. Then we have got the Huntington's disease, which is HTT gene that is mutated. Then we've got cystic fibrosis. What is the gene? CFTR, right? Cystic fibrosis. T is transmembrane, R is regulator gene that's mutated. Okay? Right. Last, we have also basically focused on the advantage of doing genetic screening, because after you get information, there's so many decisions you can make out of that and be prepared. Correct? Okay. Okay, so that's all for today. So stop recording.